thanks for sticking around for the last day during our normal lunchtime. Appreciate it. So many of us in this room are interested in biodiversity, so just imagine that slide. And I took all the photos. Before we can start to describe and understand all this diversity, the first thing we need to answer is how do we describe the population differences in the first place? So let's just imagine you have a common study system like cobras, and those are represented by these anatomically drawn snakes here. And they're mostly dark, kind of green, gray colors. And then you have another population where they're much more brightly colored. And so we might start by describing such things as length, body color, venom composition, resemblance to a chair. And we would start by thinking about the mean differences between these populations. What potentially of equal or more importance is how traits correlate or covary together. So in this case, the darker ones tend to be larger, and these tend to look more like IKEA chairs. So we recently proposed phenotype networks as a useful way for showing and visualizing these trait correlations in any particular system to generate hypotheses, and using common network metrics you can compare across systems and different taxa. And so what I'm going to be talking about today extends that to a comparative framework. So let's just start with 8 morphological traits, common things, body size, number of heads, and we'll just represent these by squares. So these would be nodes in a network nomenclature, and you might expect things such as mass and body size to covary with each other. So we'll represent that with a line connecting the nodes or an edge. And the traits are going to vary in how much they correlate with each other. So you wouldn't expect, for example, number of eyes to increase as body size increases. That'd be weird. So how does selection affect the phenotype network? So let's just imagine that one of these traits is under strong selection for exaggeration. So you might expect that if these correlations are static, that these other 3 traits that are actually not under selection get dragged along, and so the whole cluster gets exaggerated through correlational selection. However, the correlations themselves also might be the target of selection. So you might expect that if these traits are all initially weakly correlated with each other, and they all confer some slight advantages, that selection should favor overall greater integration or modularity, and so they're all exaggerated and more tightly clustered. You might expect the opposite to be true, however, if these other traits are not advantageous, and this is the only trait that confers any kind of fitness benefits, and so selection should break down these correlations, and you would have lower integration, and this one trait is allowed to freely evolve. So I'd like to ask the more specific question of how divergent selection affects signaling system, particularly sexual selection, affects signaling structure system architecture. So we looked at barn swallows. They're easier to work with than cobras, and their breeding distribution is across the northern hemisphere. So there are 6 subspecies. I'm showing the 4 that we studied for this work, and there's pronounced phenotypic variation across subspecies, despite the fact that they've very recently diverged and have very shallow genetic differentiation. So just to point out a few things, so you have these subspecies that are very pale in color compared to these that are dark, and then subspecies with really short tail feathers or streamers, and those with really long streamers. And there's increasing evidence, experimental and correlational, that these differences are due to differences in female mate preferences, and that divergent sexual selection is driving these phenotypic differences. So just to show a recent example, we studied these 2 subspecies. We looked at rustica in Czech Republic and transitiva in Israel, and we show that in Israel, where there's much darker, so this is lower body brightness, so the body is overall darker, as you can see in this cartoon, and this is concurrent with directional selection for darker breast and belly color in this population, with stabilizing selection for brightness. In contrast, we see much longer tail streamers in the Czech Republic, compared to Israel, and there's directional selection for streamer elongation in that population, and none was detected in Israel. So there's growing evidence that phenotypic divergence in this system is driven primarily by sexual selection, but it also allows us to ask the question of how such divergence should affect the signaling system architecture in general, so how all of the traits correlate with each other. So to look at this, we looked at 14 different phenotypic measures. So we looked at morphological traits, wing length, and tail streamer length, and then hue, brightness, and chroma for throat, breast, belly, and vent. And we looked at 8 populations within 4 subspecies out of the 6 that have currently been described. So we looked in the UK, the Czech Republic, Romania, Turkey, Israel, Taiwan, Colorado, and New York. And so in each of these populations, we measured all the traits, we generated correlation matrices. And so, as you might imagine, when you generate a network from this is an unfiltered network, you have a lot of edges which are most likely spurious. So we basically want to focus on the more robust edges that probably reflect meaningful correlations. And so to do that, we tried a few different ways, but for this study we used PCIT, or partial correlations with information theory, and this is a method that was first developed for gene co-expression networks, and it's very efficient, and it's in R, so that makes it great. Bootstrapping produces similar results, but it's very intensive. And so the filtered correlation matrix would look something like this, and we'll just start by looking at the network. So this is for Colorado, and so triangles here represent color traits or measurements, and squares are morphology. So this is our streamer length and wing length, and as you might expect, these are both feather length traits, so there's a strong correlation here, although it's weakly correlated to the rest of the network. So these color traits are all kind of, to varying degrees, correlated with each other. There's a lot of variation in the structure here, so again, to orient you, if you're not used to staring at these things, the thickness of the line represents the strength of the correlation. Thicker lines, stronger correlation, but the sort of orientation is somewhat arbitrary and based on minimizing the edge crossings. There's a lot of variation here, and you can see that in particular, so here I have these are the color traits, the triangles, and the dark red are throat. And so as you can see, there's these triads here that seem to be somewhat removed from the rest of the color traits, and throat also tends to load on separate PCXs, so it suggests that throat actually might be a separate signal and seems to be possibly evolving in a different way from the rest of the cluster. So for now, I'm going to focus on the body, the main cluster in these networks from here out to look at integration. And so just to remind you, so we're basically interested in this question of like how divergent selection on body color could drive integration or positive integration or negative integration. And so to encapsulate sort of an integration index, we calculated mean edge weight out of the number of possible edges. So a higher value should be stronger correlation, stronger clustering, higher integration. And a darkness index. So this is just mean body chroma for the 3 body patches. So breast, belly, and vent, and higher would be darker. And so you might expect that the traits, if the body darkness traits are initially weakly correlated, they're sort of redundant, you might expect that selection would favor increased redundancy and increased exaggeration so that it's more effective as a signal. It's easier to perceive, it's more apparent, and it's more redundant. You might find the opposite though if just breast color is important and it's costly to maintain these other patches, and so selection should favor exaggeration of just that one trait separately from the rest. And you might expect no relationship, potentially, if these correlations are really tightly constrained or they're just not subject to selection. So what do we find? We we found a really strong positive correlation. So these points are color-coded, so Taiwan's the lightest colored on our darkness index, and the 3 darkest subspecies have the highest integration index. So another way to look at this is by ordering so the networks were previously ordered west to east, so now we'll order them by the amount of integration. So Taiwan is our least integrated network, so you can see that it's pretty spread out, the connections are weak and few, and this is our palest population. So then if you look at the rest, so we're kind of increasing in integration all the way to Israel, where you have this one really tight cluster, and this is our darkest bird. So this suggests that, what's apparent here from these pictures and from looking at the birds themselves is that these are really dark and these are really light. But what wasn't apparent by looking at that is that all of the traits here are also much more strongly correlated in this population. So the take-homes from this are that phenotype networks are useful for studying divergence in communication systems and beyond communication systems to answer general questions about how selection affects correlation structure. And in barn swallows, populations with the darkest colors have the strongest correlations between color traits. And this is because divergent selection seems to affect not just the means, but also the overall signaling system architecture, which is pretty general, and it would be really interesting to look at this in other systems to see how often selection for a particular patch or a particular modality affects the overall system architecture. So future directions are to add more barn swallow populations. For those who saw Liz Cordado's talk, we have a lot more populations to add to this, and so that would be pretty sweet. We're also going to be exploring other network-based measures of phenotypic integration and extending this to species of wolf spider in my current postdoc at University of Nebraska that I inhabit. So with that, I'd like to thank a lot of people who made this possible, and I'll take any questions. So we weren't controlling for genetic relatedness in this case. So there is variation across populations, but the fact that we found this linear relationship, despite the fact that there are these variable genetic relationships, I suggest that genetics isn't explaining it. And also they're very shallow divergences.
But how did you get your sample?
How did I get the sample? So we went to all the populations and measured the crap out of them.
No, did you just go to one roost, or did you go to 10?
So these are most of these are ongoing study populations for long-term studies of sexual selection in different populations. So typically, I see. So how did we?
I think it's how do you know those aren't brothers that you're collecting.
Right.
So if you had a high degree of genetic relatedness because the population all comes from one related group of individuals, you would expect to get a high degree of phenotypic integration.
So the adults, so the nestlings have really high dispersal rates. So the adults at any population level, we sampled as widely as we possibly could within each population. So I don't think that's a major effect.
Thanks, Matt.